Welcome everyone for the second session today. We have Dr. Misha Ahrens from Genellia Research Campus. Misha received his BA in mathematics from Cambridge University and PhD from the Gatsby Computational Neuroscience Unit at the University College London. He was then a Burroughs Wellcome Fund supported postdoctoral fellow at Harvard University, where he worked in Florian Engert's group, and he developed closed loop behavior paradigms and whole brain imaging methods for understanding brain function. So in continuation of the zebrafish theme for today, so Misha also uses the larval zebrafish as a model organism. In 2012, he became a group leader at the Genellia Farm Research Campus. And his work as an independent investigator there has revealed the potential of larval zebrafish for studying neural correlates of behavior in a brain-wide manner, rather than focusing on individual circuits. And this has been a huge plus for the field. So Misha will tell us more about the technical aspects of his work and also delve more into the biology of his research work. So without further ado, I hand it over to Misha.
Right. Hi, thanks, Vatsala, and thanks for having me here. It's a pleasure. And I look forward to a lot of conversations and questions. So again, please interrupt. The plan is for today to give you a background in mostly the technical aspects, starting with where the field is, and also showing you the techniques we developed in the lab that enable us to do the biology we're focusing on now. And I'll give you an example, if there's time, I think there's time, I'll give you an example of an application of the techniques to a biology question today. And then tomorrow I'll go into more depth about the kinds of questions that we're addressing and go deeper into the biology. All right, so. Okay, you can see the slides, right?
Yeah. Yeah.
Okay, I also should, so Vatsala, will you stop me or
yeah,
I don't need to pay attention to the chat, right?
No, you don't need to. I'll monitor the chat box and read out the questions as and when they are posted.
Okay. So the title of this lecture is Optical Imaging and Analysis of Neuronal and Astrocyte Activity Across the Whole Brain. All right, and I want to start with saying that I've had the privilege of collaborating with a lot of people, and I'll acknowledge the specific people during as I present, but this is really, I think, for me, one of the great things about doing science. Oops. That was a repeat of my reminder to make sure I was online. Okay, so I want to start with this statement: understanding the brain is challenging. As neuroscientists, I think we take on a big challenge. We can, it's easy in neuroscience to make superficial statements about brain function, and it can be incredibly hard to make really deep, profound progress. And so we're taking on a lot. I respect everyone who works in this field because it's a very complex system. And one reason for this is that the networks that make up the brain are incredibly large and incredibly interconnected. And what's more, in a lot of our studies, we're only observing a small amount of this network at a time. And I illustrate this in space, but it's also true in time. So we're often subsampling the data. If we're doing, for example, calcium imaging, we're not imaging at spike resolution. And when we are recording activity at spike resolution, we might not be able to record from as many cells as we want. And a lot of recent developments in optical imaging and electrophysiology have made a huge progress in making the networks more visible, both in the temporal and the spatial domain. In the spatial domain, imaging can be used to record from larger and larger volumes. And I'm going to cover some of these methods, but keep in mind that this doesn't cover all methods that have been developed. And I'm also happy to talk about things that didn't fit into the slides, and I can send you references as well. In the temporal domain, it's often a trade-off. So if we can use dense multi-electrodes to record at very fine timescale, but then we sacrifice some of the coverage, although with recent highly dense multi-electrode devices, this is also getting better and better. Okay, the other reason is that the brain operates on many scales, both temporal and spatial. To illustrate the spatial scale, certain computations can occur in molecular networks, both within cells and outside of cells. And these molecular networks are embedded within neuronal networks, which communicate within circuits. And then eventually brain function arises from interaction across brain regions, and also interactions between the brain, the body, and the environment. And this is also extremely difficult to study across scales simultaneously, although molecular sensors with imaging are making this more and more accessible. So there are lots of exciting technical developments happening that we can exploit. And one example of this is the use of calcium indicators to look at activity in neurons, but we can also look at calcium levels in subcompartments of neurons to gain access to the subcellular scale. And we can use not only calcium sensors, but also sensors for other molecules like cyclic AMP or ATP or glucose or neuromodulators. So the combination of genetics and molecular biology with imaging technology is extremely powerful for accessing these scales simultaneously. Okay, so I'm going to focus first on recording from more neurons, and this is facilitated by imaging methods as well as multi-electrode methods, but I'm going to focus here on imaging. I'm guessing you're all familiar with two-photon imaging, and this is from a paper from 2014. I think this is a little factory bulb in mouse. We can image from a bunch of cells and neuropil, and these methods have now been extended to cover tens of thousands of neurons across mammalian cortex within the last 10 years. And when I started my trajectory in experimental neuroscience, I wanted to make use of such methods, but in an animal whose brain was very accessible and small. And the reason for using large-scale imaging methods in a small brain is that fractionally you can record from many more neurons than you would if you use a larger brain. And having this coverage, capturing more of the network, allows for observing the system more as a closed system, where there are fewer hidden variables. If you miss cells, then you can consider them as hidden variables, and you might not know where a signal that the cell responds to comes from. But if you record from, say, the whole nervous system, you might be able to find the input cells through, for example, computational methods, or by combining it with anatomical methods, and you'll then know the activity of these input cells. So to do this, we combined large-scale imaging methods with a small brain. So here you can see this brain from, can you see my mouse cursor?
Yeah, we can.
Okay, cool. From here to here in a 1 week old animal is about 800 microns. So this fits easily under an objective that can image at subcellular scales. And you can see an overlay of a plane that was recorded with a 2-photon microscope with the epifluorescence image. And this granular structure represents single neurons. So how do we record brain activity? I'm going to give you an overview of a bunch of techniques that can be used, and some of these are older, many of them are very recent. So I showed you a 2-photon. Oops! I showed you a 2-photon movie just a few slides ago. Here the idea is that you have a pulsed infrared laser beam that gets focused into a point. So this point represents the focal spot. And can scan across space in 2 dimensions, say this plane, or in 3 dimensions, say this sphere. And then you can reconstruct the volume by stitching together all these XYZ coordinates. But at any point in time, the image will look something like this. In this case, the focal spot is on this snake-like object, which we assume is fluorescent, to produce 1 spot that the computer receives and then reconstructs into the volume. But you need to scan 1 point around in space. So this is fairly slow. So several groups have developed very clever methods for either multiplexing or scanning this at incredible speeds, so that, for example, you could scan them in very quick succession at different points in 3D space, or even simultaneously. And then you could get, in some sense, more than 1 spot at every time point and increase the speed. Now, and I should say this reference here is to a review article that Philip Keller and I wrote. So don't take this as us having created these techniques. This is just a reference for you if you want to read more about it. And so light sheet microscopy is another way for doing volumetric imaging. And the idea originates from 1902, so 119 years ago, which I think is a nice example of how we build on concepts that were created in times when technology was far less developed, but the scientific creativity was at equal levels. Now, they didn't have lasers back then, so they used an illumination arm to create a laser sheet, create a sheet of light from the side, and use the detection arm to image from above. And they didn't use this for neuroscience. I think they were looking at the structure of glass or something like that. But the idea is that the light sheet gets focused, but the objective is such that it's a low numerical aperture objective, so the light sheet remains thin for some extent of space. And this prevents the sample above and below the light sheet from being illuminated. And so this detection arm sees this plane unpolluted by stuff that's above and below. And this was different from previous imaging methods where the whole sample was illuminated. And so these are modern implementations where laser sheets or laser pencils are used to illuminate the plane. So this is a laser sheet implementation where a cylindrical lens can be used to create a sheet of light that gets scanned up and down through the sample. And in this case, at any point in time, the camera, the detector, sees one plane of the sample. In this case, these 2 spots, the illumination of this snake-like object and this spherical object gets sent simultaneously, and as it scans up and down, you can reconstruct the 3D volume. Another implementation is by scanning in 2 dimensions now a light pencil through the sample. in which case you build up one plane by sweeping the pencil across. And again, if you want to find the specific references to the original papers about this technique, you can look at the reference that I sent to the review article that I sent. This is also a light sheet technique, but using only a single objective. This is from the lab of Elizabeth Hillman, and this is called scape microscopy, where the light sheet comes out of the objective, the same objective that's used for imaging. So you can see the light sheet comes at an angle and the image is recorded at another angle. And so this is a very intricate optical pathway that allows for this. And can be used also in samples that don't allow for side access. For example, mouse. In mouse cortex, you could use this for imaging and has been used for imaging, and also in Drosophila. And these animals are harder to access with a light sheet that comes from the side. So this technique really allows for widespread application in many types of animals. Light field microscopy is another method. So here it's contrasted with wide field microscopy. This is in some sense the simplest form of fluorescent microscopy that we might consider, where the whole sample is bathed in excitation light, and there's just a microscope above it that records the image. And in that case, you would get an object in the focal plane of the objective in focus, and objects outside of the focal plane, like this one, out of focus, and extended objects will be partly in focus and partly out of focus. Now you can this still has depth information. For example, the fact that these spots are blurry tells you that they are either outside of the imaging plane or extended in space. And so there's some information about depth in this image, but it's difficult to decode. All we know is this is an object inside in the sample plane and it's small, and these are either outside or big. But with clever detection pathways, for example, through the use of microlens arrays, we can reconstruct 3D information, again, approximately, but with more accuracy. So we might get a reconstruction at a lower resolution than these other techniques, but in 3D using a single snapshot. So this is also called 3D snapshot microscopy. And the idea is that you design this detection pathway, and this is here, of course, simplified. There are relay optics and so on in here. Design this detection pathway to provide maximal information about the 3D structure of the sample as it is projected onto a 2D plane. And this transformation is linear in the sense that if you make one of these voxels brighter or darker, you can describe the image that is detected as the previous image before you made this manipulation plus the difference. So if you make one pixel brighter or larger, brighter or darker, you either add or subtract the image that you would get if you only would have a small object here. And so given that this is a linear process, in principle, you can invert this and reconstruct the image through a linear inversion. There are other algorithms that people actually use for it, Richardson-Lucy deconvolution, for example. And here is an example of such an implementation from the lab of Kai Wang, where a lenslet array is placed in front of the camera. And again, the optics are more complex than in the schematic, but the idea is that each of these lenslets in principle sees the sample from a slightly different angle, and allows you to then approximately reconstruct the three-dimensional sample. So you can see that here in the case of a zebrafish brain, dorsal view, side view, with activity patterns. And they use this for imaging in freely swimming animals. Okay, so we use light sheet microscopy in zebrafish, and I'm going to just show you the setup and the data that we get. So This is a fish in the sample chamber. Here you can also see the size of the small size of this. This is an objective here that I'm outlining, and this is an electrode that records signals from the tail of the fish. I'll show you later why we do this. And this is the entire extent of the animal. So it's really a small animal, and yet it performs a bunch of, I think, very interesting behaviors. And so here's a schematic of the microscope that we use. If you would visit the lab, there's now a whole bunch of extra optics inside this system for two-photon manipulation of neuronal activity and other optical systems for also perturbing activity that I'll introduce later. So this is the type of data that we can record multiple times per second. We can capture an entire zebrafish brain volume at single cell or subcellular resolution in almost all parts of the brain. So you can see that here, a sequence of images from just taken from somewhere in the brain. Some parts are slightly below single cell resolution, but most parts are above. About 85% of the brain we can get at single cell resolution. And this means more than 100,000 neurons out of the roughly 120,000 neurons we can get at this resolution. And so this technique really allows you to connect the cellular scale to the whole brain scale and look at the brain as a more or less complete system where every activity of every neuron is represented. And this was when I moved to Janelia in 2012, Philip Keller and I developed this technique, collaboration with others, and then improved it later, and that produced some of these first whole brain recordings in a vertebrate. So we wanted to, or my lab's interest is in relationship between behavior and neural activity. So we wanted to combine this with virtual reality environments in zebrafish. And this was really inspired by work that had been done earlier in flies and mouse, where labs like labs of David Tank, Kavi Memon, Vivek Jayaraman used virtual reality systems to essentially allow animals to play computer games while their brains were being recorded.
Misha?
Yeah.
There's a question in the chat box. So Bidisha asks, in the slide where you showed the whole brain activity, what is T-1?
Oh, let me see. I think it's just an arbitrary, T-1, I think we triggered on activity of these neurons. This was spontaneous activity. Okay, so the movie that you see below is not spontaneous. This animal is viewing visual stimuli, and we're also recording it's swimming. And so the activity that you see in this movie, these two bursts of activity that are being looped correspond to two swim bouts. But this was from another animal that we recorded spontaneously, and T0 is just the time of this spontaneous burst of activity in these four neurons. So it's essentially an arbitrary point in time.
Thank you.
So we were very inspired by this type of work where animals could traverse virtual reality environments, and this was just amazing back then. I'd also seen some of this done in Drosophila that was glued to a stick and could fly left and right, and the environment would rotate in the opposite direction. And so let me see if I can. So when I was a postdoc in Florian Engert's lab, I implemented this for paralyzed larval zebrafish. And the reason for doing it in paralyzed fish was, well, there were multiple reasons, but one of them was that the brain wouldn't move at all. and so imaging was very convenient. And yet we could record these motor signals from the tail using electrodes. So the fish is here. You can see the head and the tail ends here. There are four electrodes recording from the motor nerves in the tail. Typically, you need only two, and you can see this animal is navigating a two-dimensional virtual reality environment with a simulated flow. So when the animal doesn't swim, there's flow, and the audio here represents the signals from the tail, which is just an audio recording from the electrophysiology amplifier. And the idea is that these signals are fed back, are decoded to figure out if the animal wants to swim or turn, and fed back into the virtual reality environment so that the animals can actually move through it. So these animals swim in swim bouts, roughly 1 bout per second, and that's the crackling noise that you hear. All right, so to give you an idea of the types of signals that we get. So, okay, first of all, so the setup is a paralyzed fish, electrodes suctioned onto the tail, so they don't penetrate the skin. It's very much like EMG recordings in larger animals where you use an electrode pad. And this is then decoded by the computer for forward swimming or turning, and then used to traverse virtual reality environments, and can be combined with light sheet microscopy. And we use a light sheet microscope with a side illumination and a front illumination to get the tissue between the eyes without ever shining a laser into the eyes. And here's an example forward swim and a leftward turn. So you can use this precise pattern of undulations to figure out if the animal is trying to swim forward or turn. And feed that back in real time. So here's just this movie that you just saw. Now this is, on the left is a freely swimming animal performing the optomotor response. That's a response to visual motion. And the interpretation of this is that the animal thinks, if I can anthropomorphize, that that there's flowing water and it should stay in the same region of space. And a very strong cue for that is the motion of the fish relative to the visual environment, because if the animal swept backward, for example, it sees the world move forward, and so in response, it swims forward. And this animal in VR is performing that very same behavior. Now you can ask, how similar is real and VR behavior? So we checked that in the context of the optomotor response, and there the statistics were very similar. So on the left, you can see the free swimming behavior in the kind of setup that you saw on the previous slide. When the stripes move forward, the animal. So in reality, the stripes move in various directions depending on where the fish is, but relative to the fish, they always move forward. So if you then rotate all these trajectories, you can see the fish swims in the direction of the stripes, but not always. It has some deviations. And this is the same data, but reconstructed from a virtual trajectory. You can see it's longer time, but the scale bar is different. So overall, the behavior looks similar. It's not identical, and that's probably because animals are more agile when they're freely swimming, but overall they perform similar things. We can combine that with light sheet imaging. Specifically, we have these two lasers to record from the in between the eyes with the front laser, and everywhere else with the side laser. We sweep these simultaneously. So that's shown here in the diagram. We never shine the laser into the eyes. Although the fish does see some scattered light, it doesn't get, we always try to avoid getting a laser pointer in our own eyes, so we also don't expose the fish to that. And then we scan these sheets in the vertical direction, so that as the data comes in, it looks a little bit like that. It's even faster now. You can see individual neurons, and it scans through the brain fairly rapidly. So that gives you the type of data that you saw before. And as I told you, we can combine that with behavior. And we can also combine that with imaging of more than one type of cell. So this shows you two cell types in the brain together with behavior. It doesn't show you the visual stimulus that the animal sees, but this animal is also exposed to visual stimuli. So the behavior shown at the bottom, this is fictive behavior. It's essentially the signal that you heard before through the speaker, but now shown visually. This is a rectified electrophysiology signal. So when the white line shows vertical deflection, it means the fish is swimming, and when it's down here, it means the fish is not swimming. This is sped up by a factor of 2.8. Now you see neuronal signals at the top and radial astrocyte signals here. So at the bottom, the radial astrocytes are a type of glial cell in the zebrafish brain that used to be typically interpreted as kind of helper cells to keep neurons healthy, to look after tissue homeostasis and energy delivery and so on. But in studies that I'll present to you in lecture number 2, we found that they do much more and that they're actually active participants in information processing, working in tandem with neurons. So here the data is collapsed over 5 slabs. Each slab represents multiple planes that were imaged. So each slab represents 25 microns of the brain. And we took a maximum intensity projection across every slab. So this is now neuronal activity, glial activity measured through calcium, behavior, while the animal is traversing a one-dimensional virtual reality environment. So you can imagine that this produces data sets that are kind of big, not trivial to analyze. Just first of all, because of the size, you have to extract tens of thousands of cells from this and then try and interpret them. So I'm now going to cover some of what to do with the data and to really go from experiment to data to analysis to an understanding of the data. I think there's a huge space for creativity here, application of computational methods, machine learning. There's another question, Vijay?
Yeah, Misha, if you go back to the previous slide, the 1D graph that you showed, I didn't understand. I mean, the signal that you get is both a spatial and a temporal signal, right? So
this one here?
Yeah.
Yeah, so the spatial signal consists mostly of 2 channels. On one side of the, let me go back, it's essentially this. So it's the left channel and the right channel, which we can then algorithmically turn into a distance and an angle. So there are various ways to do that, and we tested various decoders of these signals. But this, where the left and the right channels are more or less balanced, it represents a forward swim. Whereas here, where the left channel is a lot stronger at the start, represents a left turn followed by forward motion. Is that what you mean?
Yeah, but in the next slides, you had a 1D signal. There was an up, down.
Yeah, so we simplified it. And the reason is just because otherwise the movie gets a little rich. But also because the animal was swimming in an environment where the visual motion was mostly forward or backward if it swims in closed loop. And you can see in this here movie, oops. Okay, I'm just going to turn my sound off so you don't have to deal with the noise. But that means for a few seconds I won't be able to hear you. Yeah, 1 second.
Sure.
So you can see that when the motion is parallel to the axis of the fish mostly swims forward. Okay, so my sound's back on. And so we assumed in the movie that showing both signals, just summing both signals, or rectifying and then summing both signals is a good enough representation of behavior in this 1D environment. Does that make sense?
Yeah. Thanks.
Okay, but you're right. it will also have turns. Oh, and actually, you're even more right, because we know in this case, I'll explain tomorrow, that the animal is trying to turn. So this is very much a reduced representation of behavior. There's indeed much more in here. There are turns, there are potentially also some very vigorous swims that are not well represented. So this is really just a summary.
Thanks.
All right, so then we have all that data. We need to turn that into an understanding of the biological processes that we're witnessing, which is not trivial. So during my PhD, I actually worked on neural encoding models in auditory cortex, where maybe 2 years I worked on analyzing single unit activity, trying to understand how spikes in auditory cortex arose from complex sounds. And very simple models included linear filters that could be applied to the spectrogram of the sound. Those were found to be insufficient for understanding what these single neurons were doing. So I worked on machine learning methods for describing more nonlinear models that could capture more nonlinear phenomena. And those were successful, but still didn't quite get us all the way we wanted, where we wanted to be. We couldn't still predict the neuron activity with 100% accuracy, but we got a lot closer. Now, those models were applied to individual neurons, and now these data sets suddenly had 100,000 neurons per fish. So how do you turn that into an understanding of what all these neurons are doing, and even more how they are interacting? So around the time that I started at Janelia, Jeremy Freeman also started here. Interestingly, he's now working on climate change. He really changed focus after using computational methods in neuroscience. He went on to applying computational methods to climate change. But while he was here, he developed methods to perform analyses on compute clusters using parallel computing. So he created a software package that was built on the Spark system for parallel computing to create methods, kind of turnkey methods where the goal was to have a kind of MATLAB style interface or Python interface where you could run algorithms on data relatively quickly. And so he implemented techniques for dimensionality reduction, clustering, fitting tuning functions, performing linear regression, summary statistics, and so on. And these algorithms are still in use, although since then a lot more advanced methods have come out for parallel computing. But here's an example of an analysis that he did. So this is brain activity that arises from the animal viewing these directional motion stimuli. So these are averaged across trials, but you can see that every angle of motion gives rise to a different pattern of activity in the brain. And these are, of course, mixtures between purely visual responses, motor responses that actually make the fish swim and turn, and intermediate responses or signals that are fed back from the motor system, perhaps the spinal cord or premotor centers as efference copies. But you can do a very simple thing and just ask how does brain activity depend on the angle of the stimulus? If you color the angle, you can get volumes like this and find that, for example, in the hindbrain, in circuits that we know are more involved in motor control, you kind of have a clean segregation between left and right tuning, whereas in this visual area here in the tectum, it's more intermingled. The visual tuning is more intermingled. And so, and I want to emphasize that we still get single cell resolution in most parts of the brain. So this really allows you to look at large scale activity at the same time. You can perform dimensionality reduction on such data to look, for example, at representations that depend on angle on the right and representations that depend on time on the left. And this is PCA done on the about half of the brain. This is the tectum, the cerebellum, the hindbrain, other parts of the hindbrain, and so these are just example methods that you can use for. I would consider these to be more or less a survey into what these cells might roughly be doing, but not yet. I wouldn't call this an understanding it. So I'm just seeing the slide. Oh, what are we looking for? So looking at. So this is. Maybe I can motivate it with this slide. So are you all familiar with dimensionality reduction methods like PCA or clustering? Okay, good. So so this is what you see here is the data projected onto the first principal components, and this is then a maximum intensity projection from the top. So you take the first principal components and you color code the coefficients in an angular representation. So this is the if you every voxel in this data, you can interpret the projection onto PC1 and PC2 as coordinates. You can turn that into an angle so that you lose the information about amplitude, but you keep the information about the relative tuning to PC1 and the relative tuning to PC2 in time. And so here you can, at 0 degrees, it's purely the first principal component, and at 90 degrees, it's purely the second principal component. And you can color code them according to the proportion to which they are described by these, one or the other. And this is then the temporal specificity. And you can see, for example, that this region, this is the lateral medulla, is in antiphase with most cells in, for example, this posterior hindbrain, because they have opposite angles, and these are indeed in antiphase if you look at the temporal movies. And this is now direction specificity that you saw before. This is just a tuning curve. So that the color here represents the angle that maximally activates those neurons. And this, so that was a dimensionality reduction to 2 dimensions, and this is the same thing. So here you can this is not quite PCA. I need to actually, it's been a while, I would need to look up exactly how we did this. But the method led to the time trajectories being maximally represented here. So you can see it's more or less a loop as these trajectories traverse in time, where you can tell by where the cells, where every trials is, sorry, every white dot here is one trial. And if you know where the white dot falls along this circle, you can tell where in one of these trials at which time point we're looking. So this is one trial. Here's the next trial. Yeah. So so the point on the circle tells us where we are in time, and here the point on the circle from a given trajectory tells us where we are in angle space. So which angle did the stimulus go through? Wouldn't the errors for the time trajectories be large given the kinetics of GCAM? It's not too bad because the movie was sped up. So I think one trial takes about 10 seconds, and the time scale of GCAM is on the order of 1 second. So there would be some error, but I would think it's relatively small relative to the time scale of the stimulus. The most important thing here is that every point here represents the entire brain, and every point represents one trial. So we can do a trial by trial analysis by collapsing whole brain activity into a point in 2D space. and representing time as trajectories. This is just a very simple example of dimensionality reduction. You can do much richer things, like this is from the labs of Georges Desbrejas and Bernard Englitz, that took data and used a restricted Boltzmann machine to have a more network computation level description of whole brain activity. And I wouldn't say I'm qualified to go into too much detail here, but the important point is that here we get connectivity between observed units and hidden units. And sometimes principal component analysis you can also interpret as connectivity between observed and hidden units, but it's more abstract. In this case, it's more explicit. We can look at these at the structure of the models that arise from these fits, the temporal traces that are associated with the hidden units that were fit, and then also map it back to space as you saw before as well. So this would be a temporal representation here of the data, and this would be a spatial representation of the data, where by mapping back in some sense the basis functions for these hidden units, or the receptive fields as they call it, of these hidden units, we can look for functional structure in the brain. And the nice thing is that because we have completeness, that you can recapitulate both functional and anatomical clusters. So some of these clusters, sorry, I'm distracted by the, let me finish this slide. Some of these clusters correspond to, for example, clusters of cells that are can be genetically labeled. But is it okay if I read the chat, or do you want me to close the window so that you can
yeah, you can close the window, Misha. I'll find a suitable time when you're between slides or something to ask it, so that you're not losing your train of thought.
All right. Okay. Anyway, I think now would be an okay time to ask it.
So Alok is asking, I find the lack of activity between stimulus presentations quite striking and rather surprising. Aren't there neurons which are activated by turning off the stimulus, for example, or neurons that don't care about the visual stimulus presented at all, or maybe even spontaneously active neurons? Am I missing something?
That's an excellent question, and you're entirely right. So I think we took this data with an older calcium indicator, where the signals weren't as apparent. I'm not sure why in this movie the brain is so quiet in between stimulus presentations. Let me see if I have a movie of spontaneous activity. Actually, I don't, but there is a huge amount of spontaneous activity going on in the brain. I can include such movies later. So I think it's probably the indicator being being not as sensitive as we'd want. It could also be some of the processing, because we were constantly showing stimuli. We might not have got a good baseline. So since calcium signals, if a neuron does nothing, calcium signals are non-zero. So typically you try to introduce a range of activity in neurons, and then take, say, the minimum activity that you observe as the baseline. And it could be that as we did the processing of the data there, we didn't get a good baseline estimate because we were almost constantly showing these stimuli to the fish. But you're absolutely right. There's a lot of spontaneous activity, and you're right in flagging that that was missing from that movie. But please don't take that as representative. Okay, cool. All right, so so this is one more advanced analysis that people have published. Here's another one. This is called raster map by Carson Stringer here at Genelia in collaboration with Marius Pachitariu, which is a clustering method where you can take activity here. There are about 58,000 cells. This is data from the lab that they use to demonstrate their method, and it allows you to cluster in a clever way and also sort the activity traces to kind of maximally represent the different types of dynamics that are present in the brain. And so, and here, I don't know exactly where, but there are periods of spontaneous activity in here. And you can see that the brain is actually never really quiet. My guess is that given the lack of periodicity in this period, that this might be a period of spontaneous activity. You can see that some cells are quieter than others, but the brain is definitely not silent. So raster map gives you clusters, sorts the clusters, and so by color coding them, you can visualize, as before, the different patterns of activity across the brain. And so again, typically you get multiple representations, right? All of these things represent brain activity over time, but in different ways. So I think this is a very rich field to continue to make progress, also by combining these types of representations of the activity in the brain with other information like cell types and connectivity. We can do a lot of exciting things by intersecting these very different data modalities. And here's one example. We can try to interpret all this activity in the context of brain anatomy. And this is a very active field as well, where a lot is happening. So here you can see a representation of Z-brain that is, this is this brain is upside down. This is a group that took data from Z-brain, which comes from Florian Enger's lab, and then created an interface. They need to flip this time axis. But here you can, it's a good demonstration of how different brain regions are identified in the larval zebrafish brain. And so we can map our functional data to atlases like this to get information about, to get labels for the different regions that we observe. This is from Max Planck, from the lab of Herwig Bayer, where they have an atlas that labels brain regions, genetic markers, so specific genes that are expressed in different parts of the brain, and also neurons. They traced the morphology of individual neurons, about 2,000 neurons. So this is a subset of the brain, but it does give us very good hints about how brain regions can be connected to each other. And you can, they've used that, for example, for tracing neurons in the tectum and to find that they have projections to the hindbrain. Electron microscopy is another very promising, a very promising technique for getting real ground truth connectivity. And this is a volume from the Engers and Lichtman labs this data is not being used to reconstruct connectivity, but other more finely imaged volumes from multiple labs are now being used to reconstruct the connectivity between neurons. So functional activity across the zebrafish brain is not all we have, and I think we need structural data as well to really figure out how these circuits work.
Misha, may I ask a question?
Yeah.
These rich data sets, right? I mean, from every fish, you're going to generate such highly dimensional data sets from different regions of the brain, and then multiple trials, different kinds of visual stimuli, and so on. But then there's also the added problem of having many different fish model preps from which you do the experiment. So how do you combine data sets from multiple fish to generate some sort of an understanding where there can be, individualistic differences in how the brain works?
Yeah, that's a great question. So one way to do it is kind of superficial. You sort of get these get an idea of these functional clusters in different fish, and then you average them, or you find some statistical method to combine them across fish. For example, you can find statistical criterion for asking, in this part of the brain, is there significant kind of hits across fish that are statistically significant and repeatable, so that if I record a new fish, I can make a prediction about the probability that, say, within 10 microns of this particular cell, I can find another cell with that property. So that's one way to do it. Another way is to use genetic or anatomical information to kind of further constrain the cell type that you're looking at, and not just go by location in the brain, but actually also go by the type of cell that you're recording from. And then you can also imagine computational methods of finding a kind of ground truth computational fish that can map onto individual fish. That's also something we're aiming to do. But it's definitely
like a
non-trivial problem.
functional standard brain, just like an anatomical standard brain.
Yeah, exactly. But then everything I just said is about kind of finding commonalities across fish, right? And you also mentioned looking at the differences across fish. And I think that's very interesting. You could look at deviations of individual fish from kind of the standard fish, and then finding out how this maps to differences in behavior. This all, of course, is in closed loop with behavior. So if there are behavioral differences, they're likely differences in brain activity. And if there are differences in brain activity, I think oftentimes there'll be differences in, this will be reflected in differences in behavior, depending on how finely you probe behavior. So yeah, I think it's a very interesting problem, and also not an easy one.
Thanks, Misha.
Okay, thanks. So the other thing is, I showed you how to record brain activity, but it's optogenetics, of course, gives us great power to also perturb brain activity and start looking for causal relationships. And this interfaces with connectivity, because most, or a lot of functional relationships or mechanistic relationships between neurons will be mediated by synaptic connectivity. Not all. There's volume transmission of neuromodulators, for example, is not captured by that. But in any case, just the imaging data I showed you doesn't directly capture causal relationships. You can try to infer them with computational methods, but we wanted to infer them more directly. So the idea between this part of the work in the lab was to first record from the brain and record behavior, and then map activity using any of the methods that I just described, but then and create models for how these systems are interacting, and then perturb them in the same fish in the same experiment, for example, through 2-photon optogenetic activation or through 2-photon ablation of individual neurons. So you can imagine, based on activity mapping, that you might hypothesize that two sets of neurons are connected, and that activity in one region depends on activity in another. Then you can start messing with the hypothesized upstream activity, and then re-image the brain and look at behavior to see if that relationship has changed. And our goal was to do both single neuron ablation and single neuron optogenetics. For this, you need to analyze whole brain activity kind of on the fly. So we combined a light sheet microscope, represented here by this vertical pathway and these two horizontal pathways, but also add in a system for 2-photon ablation and stimulation. And it's more complex than shown here, especially for the 2-photon optogenetics, where we have to descan the objective movement with an electrical lens in this pathway. So this interfaces with the acquisition microscope and control system. which we then hooked up through a fast connection to the compute cluster so that we could record activity and then analyze it on the fly during an experiment and then feed it back into the ablation or stimulation system. So here, an example where this is based on just 10 minutes of data, we can estimate for every voxel in the data set whether it's tuned to swimming in red or forward visual motion in green or backward visual motion in blue. And then based on this, this is Nikita Vladimirov. He built an interface for MHJ where you can select cells by hand. You can also imagine doing this automatically, for example, by interfacing a clustering method and automatically detecting the neurons that you want to ablate. So here he just ablated a cluster. He just selected a cluster for ablation, which we can then ablate. So here are 4 little regions of the brain represented as functional maps. Here is now we can image them first and then ablate them with single neuron resolution. They become very bright because we target the center of the cells. So this is a nuclear GCAM marker. It will cause intracellular calcium increases. And that's why the neurons become so bright, but they actually are then ablated. If you rederive the functional map, you can see that the neurons nearby are still functionally tuned to the different stimuli or to swimming, but that the targeted neuron is now dark. Then afterwards, we can ask what happens to the brain. So here are some example clusters that we targeted. Again, this is a pre-ablation functional measurement. And this is now the difference in brain activity after ablation of these 2 clusters of cells. So there's, of course, a decrease in activity in the clusters of cells that we ablated, shown in green. But you can also see there's a difference in activity widespread in the hindbrain. And that's presumably because of connectivity, where a decrease here leads to an overall decrease in motor circuits. You can do that for these different clusters. And in this way, we can start causally interrogating this system. So I think I'm out of time, right? I had a biological study as an example of how we can apply these techniques.
We have till 8:30, Misha. Since we're taking questions in the middle, you can go until like 8:20 or something, and then we'll take questions in the last 10 minutes. We're also taking questions in the middle, so it should be okay.
Okay, so it started at 7. Wait, it started at 6:30, right?
7 local time, and
so sorry. Okay, so I have another half hour, right?
Right. Yeah. Yeah.
Okay. Okay. So I mean, we could also have a discussion, but I can also. So you're suggesting I continue, right?
Yeah. Why don't you continue? Yeah. And then if you finish at like 8:20 or something, then we can have more questions at the end.
Yeah. Okay.
Yeah.
Okay. So the neural basis of exploration. So this is a study we did more than 5 years ago, but it illustrates how you can combine these methods. And then next lecture, I'll go into much more recent work. Okay, so this is a behavior that we observed in freely swimming animals. So this is the behavioral work was led by Tim Dunn, who was a student at Harvard, and the imaging and optogenetics by Yumu, who used to be a postdoc in my lab here at Janelia. So it's a collaboration between the groups. And what Tim found was that when larval zebrafish swim spontaneously, they concatenate turns that go in the same direction. So you can see turns to the left in green. The animal often makes more than one green turn, and the animal often makes more than one magenta turn. When we saw this behavior, it was interesting because of the time scales that are involved. the timescales are about 10 seconds, which is of course much longer than the timescale of a single neuron firing, a single neuron spiking, and suggestive that there's some higher order structure to the behavior that might be useful for the fish in exploration. So these fish are just spontaneously swimming in a featureless environment. And what an animal needs to do in a featureless environment is to find out what is out there, for example, food. And so this could be an efficient foraging strategy. We actually simulated later on that indeed this is an efficient strategy. And there's even a paper, an engineering paper about a robot that has a chaotic oscillator to drive exactly this type of exploration for the purposes of more efficiently and more evenly covering a space when you're exploring it, when there are no olfactory or visual cues available to you. So to show you what this looks like in a swimming fish, this is, I think this is the same trajectory as you just saw, or similar one from another fish. And this is the same thing in virtual space using the type of virtual reality techniques that I just described to you. You also get these correlated turn sequences. And we wanted to know how the brain does this. And so here you can see spontaneous activity. So this also answers the earlier question about why is it so quiet in between by Alok. Why is it so quiet between stimulus presentations? So it's in fact not. If you do it more carefully, this animal is just presented with a red screen with no stimuli on it, and it does swim. But you can also see that when it doesn't swim, there's a lot going on in the brain of this animal. And so we figured that somehow the brain must be generating these turn sequences. And our initial hypothesis was it might be the forebrain. So this is a region. So the pallium, for example, is a region that has the evolutionary predecessors of the neocortex, the hippocampus and the striatum. And so we thought this higher order behavior might arise there. We were wrong, but we did figure out where it does arise. By the way, another thing we can discuss is a bit of neuroanatomy, just to give you an idea of how this maps onto mammalian brains. I just described the pallium, the olfactory bulb is up here. Here's the habenula. This is the homologue of the superior colliculus. It's called the optic tectum. The cerebellum is here. It has the same cell types, Purkinje cells, climbing fibers coming from the inferior olive, that's here. But on the output side, instead of the deep cerebellar nuclei, it has uridendroid cells, which are intermingled with the Purkinje cells. And the uridendroid cells are the homologue of the deep cerebellar nuclei. So there are a lot of commonalities, but also some differences between these vertebrate brains. And there's a huge amount more to delve into. There are labs that exclusively study the neuroanatomy, and it's a super interesting subject. We're always trying to catch up. Okay, so, but coming back to this, how do we analyze this? So this is an analysis that Jeremy Freeman did based on the tools that he had developed. The fictive swim signals from the two electrodes are turned into an amplitude and a direction. So the amplitude is kind of how far the fish swims, then the direction represents how by what angle the animal wants to turn. And so this also addresses the earlier point of that these signals are actually not one-dimensional. So Jeremy mapped them to a two-dimensional space representing kind of the pad that you land on here. This is a two-dimensional space, but they're 12 basis functions representing does the animal want to take a strong left turn or a weak left turn or a strong forward swim and so on. Convolve that with a calcium impulse response function to get regressors that would be similar to calcium traces if the calcium traces literally represented this behavior. These regressors can be then turned into receptive fields using linear regression. So this is just a visualization of the coefficients of this model, where you're trying to predict neural activity from these regressors. And then you can predict neural activity. These are two example cells. And show these example cells as a function of left or right turns. And then he collapsed again this receptive field onto just a laterality index from left to right, which could then be mapped back to the brain. So this is so here's an example of a top view, a front view of the brain, and two side views, the left and the right side of the brain. You can see it's relatively consistent across animals, and always this structure shows up in the center, in the anterior hindbrain. So this we call the hindbrain oscillator. We originally called it the hindbrain oscillator when we were analyzing spontaneous brain activity, but without knowing behavior. After we also recorded behavior, we changed the name by suggestion of a reviewer to the ARTR, the anterior rhombencephalic turning region, which I kind of regret because it's an ugly name. But the reason we did that was to combine an anatomical label with a functional label that it's involved in turning. There are also cells in the inferior olive here, and we believe they're directly connected. Here's a visualization where you can see the tuning to forward swimming and the tuning to turning. That directly come out of these analyses. And this is now one of those maps where we combined data from multiple fish and looked for consistency. So this is this addresses Vatsala's question a little bit, but in a very crude way. This is in some sense just an average. And you can also see superimposed on this volumes from the Z-brain atlas. So here, these cells are the reticular spinal system that projects to the spinal cord. Here is anatomical information. These are the rhombomeres, and here are a couple of different brain regions, just as an example. And one of these brain regions down here is the inferior olive. So you can see that by mapping these data back to anatomical information, we can get regional specificity. And so here's activity. This is sped up, I think, by a factor of 10. This is not the same fish, but just the hypothesis that we were testing whether activity in the ARTR, namely this region here, is indeed underlying these swim patterns. And the reason is because of the strong correlation to the turning direction, but also the slow alternation between the left and the right activations that you can see in this functional image, in this functional movie, that occur on the same time scale as the left and right alternation in the behavior.
Misha, there's a question. Sriram asks, how do we go from these brain-wide activity maps to how circuits generate them?
Yes, that was exactly our question. So for this, we can then apply the optogenetic methods. And again, as I said earlier on, in neuroscience, there's a great deal of depth that we could go into. And I have a constant sense of restlessness because I always feel like we're not going deep enough. But in this case, so although we don't have ground truth connectivity yet, we had a very good idea about how these neurons were steering in behavior, which we first tested by combining functional imaging here shown in one plane with optogenetic stimulation. So we functionally image these cells, figure out what they do using a quick online analysis, and then stimulate them optogenetically with a 2-photon microscope. That's all built into the same system. And when we stimulate a couple of cells, less than 10 on either one or the other side, we can bias turning into one direction or the other direction. You can see here, for example, fish, consistently when you stimulate the left side of the brain, the fish will turn more to the left, and if you stimulate the right side of the brain, the fish will turn more to the right. You can see that this turn bias outlasts the period of stimulation by several seconds, consistent with the long time scales that we see in the behavior. We also did the opposite experiment where we ablated either one cluster on one side of the brain or the other cluster on the other side of the brain. And there we found that we could bias swimming away from the ablated direction. And here the analysis was more tricky. And the reason for that is because if a fish has no memory of what it did before, it'll still have some statistical chance of turning in the same direction, namely 0.5. So we needed to kind of filter that out. That's why I didn't include it, but if there's time at the end, I can pull up those slides as well.
So there's another question. Sonia asks, have simpler brains where circuits have been worked out and whole brain imaging is possible, been able to achieve that? By that, I think she means circuit-based understanding.
Yeah, so to some extent, but again, we come to the issue that neural computation occurs on many scales. So in C. elegans, you can do whole brain imaging. The brain has a little over 300 neurons, and the connectome is known. Of course, there's variation across animals, but it's fairly well, you can fairly well approximate connectivity. But there also, when you try to build a model worm, and there's an open source project where people try to build model worms based on information that they've collected, but you find that there if I understand the state of the field correctly, it's also not possible to generate all the behavior with a simulation, because it depends on very intricate aspects of the brain, like gap junctions or the dynamics of synapses, the volume transmission of neuromodulators, and so on. So because of the multi-scale nature of neural computation, it's very difficult to do, but we can get to some insights. For example, in this case, we could establish a causal relationship to behavior. We have a very strong hypothesis for how these cells are connected to one another to create these long time scales, and how they're connected to motor systems, for example, through the inferior olive, that we'll be able to check when the connectome becomes available. We can also look at the anatomy in more detail. So this is functional imaging combined with markers for glutamate and for glutamatergic neurons and for GABAergic neurons. And from this, here we can take this top one as an example. You can get functional information, map that onto the anatomical information to find that the center clusters are glutamatergic and the side clusters are not, and the side clusters, in fact, are GABAergic. It's a little hard to see, but these are labeled. And so we can now identify the neurotransmitter type, and we can do preliminary tracing through activation of photoactivable GFP to see that these GABAergic cells project to the other side. And so now we have a direct hypothesis for a circuit diagram of how these are connected, where we think that these clusters are connected within themselves, either synaptically or through gap junctions, that could give rise to these 10-second timescales, and that they inhibit each other through these GABAergic clusters. We can use that to simulate this behavior using a model where the hindbrain oscillator is connected through mutual inhibition. So these are the ARTR, or hindbrain oscillator, and then these activate premotor neurons that project to the hindbrain, and then feedback is received back into the ARTR. And this is now also reflected in real activity. After a turn, the ipsilateral, this uses the old label, the ipsilateral cluster is activated after a turn and then very slowly decays. which we modeled as feedback from the fish actually making these turns. Another group, the group of Sharad, they found that actually we got this wrong, and it's actually the GABAergic clusters that inhibit the contralateral premotor circuits. So rather than encouraging the fish to turn left, the circuit seems to be preventing the fish from turning right if the fish has turned left before. But this decays, and so this memory doesn't last forever. And there's, of course, some stochasticity and inputs from other brain regions feeding into these circuits. And that's how the fish will be biased to perform the same turn, but will not get stuck in that mode forever. And this model is able to recapitulate the behavior very well, where you can see the model also makes multiple turns in one direction and multiple turns in the other direction. And the activity of these model clusters looked quite close to the real ones. So this is kind of how we try to patch together all these different techniques to look at the circuit from different angles, functional, structural, molecular, and gain a circuit understanding of what's happening. We can use statistical techniques to describe what to interpret all this data in a more engineering framework, where we think that this system is essentially implementing a two-state Markov model to give rise to correlations, where a correlated fish has a different type of behavior from a random fish, where a random fish means a fish that picks a left or a right turn randomly at every moment in time, independent of what the fish did before. And I just fly through this, but we also tested these different model fish for foraging efficiency, and we found that depending on how you constrain the random fish, you get more efficient foraging in a fish that has these correlations between subsequent turns, or is more efficient in terms of energy expenditure, because turning across a large angle costs energy. So you can interpret this also in a framework where animals are trying to optimize their behavior for spending less energy and still covering the environment efficiently in search of resources like food when there are no cues available. All right, so in general, our goal remains to connect kind of algorithmic understanding represented here by this two-state Markov model, which we use for interpreting the circuits and the behavior, with circuit level understanding and behavioral level understanding. And so in this context, tomorrow, I will give you a more in-depth discussion of recent research from my lab, including on neuron-glia mechanisms for brain state switching and on a brainstem integrator for self-localization and positional homeostasis. And as I said before, it was really wonderful to be able to work with all the people who contributed that I'm showing for the particular projects that I showed here, include Nikita Vladimirov, Chen Wang, Philip Keller, Timothy Dunn, Yumu Sujata Narayan, Jeremy Freeman, and Florian Engert. We also work with a lot of other people in the lab as benefits from a lively environment where a lot of people help each other. The work I showed you benefited from others as well. All right. I also put my contact details if you want any follow-up discussion, but I'll also be back tomorrow.
Thank you so much, Misha. So there's also a Slack channel. I think the office has sent a link. So, if you can if there are
just
questions.
Now or earlier?
Earlier. It should have been in the mail. If not, I'll forward it to you. I have it.
Okay.
but if there are questions, people can ask. But in the meanwhile, I'm just wondering, so you have this, for any behavior, a region that's involved in it based on these whole brain maps, right? For example, let's take this ARTR or the HBO. Even though, when you ablate it, you lose the behavior. How exactly, in a conceptual framework, if you're venturing into this anew, how do you rule out upstream contributions, or that there are other regions that may in fact be instructing, let's say, the ARTR or the HBO? For how do you rule that possibility out?
So certainly it receives input from other regions, for example, from premotor regions or maybe in the spinal cord, and we have strong evidence that it also receives visual input and others. So no circuit works on its own, right? So and part of that is the way we analyze the data. We focused on correlations to the most prevalent aspect of behavior that we were focused on, namely turn direction. And so we get the cells which have strongest correlation to turn direction. But by no means is any circuit autonomous. And so for that reason, these methods like restricted Boltzmann machines or other statistical methods that consider all activity across the brain simultaneously are very important. So generally, we've been focusing on kind of the central circuit, the sort of the circuit pattern consisting of maybe 3 cell types, but we always know that they receive input from a huge amount of other cells and also project out to a huge amount of other cells and not just behavior. So that's an example of kind of the continued restlessness that we all feel because we know that we're not capturing everything, even though we're recording from everything. So historically, we've been good at kind of pinpointing the most central regions that are involved in behavior, but we're actively working on including all the other parts. But that requires either very good machine learning tools, frameworks, or very strong information about, for example, connectivity across cells, as we're hoping to get from the connectome, or molecular information. So there's always, so you're absolutely right, and we're always trying to improve on that.
No. So maybe I should be a little bit more provocative and ask you. So you have this very powerful technique, right, which is to look at the whole brain during any given behavior. So how do you think about behavior being generated as from these, you say, okay, ARTR controls or makes the decision for a turn behavior, or would you think about it in more of a loop kind of configuration where there are inputs and outputs coming into specific regions, and therefore the computing is more distributed? These are, two different ways of thinking about how an organism makes a decision to do something, right?
Yeah, so completely in a loop framework. So also keep in mind that we kind of reduce the environment to a featureless environment. So the animal is completely relying on self-generated activity when performing its behavior, but there's still a loop through behavior back to reactivation of these cells. So these cells bias swim direction, but they don't directly drive swimming. They only bias the swim direction when other circuits decide to swim in some ways. So this is not the swim generator. This is it biases swim direction, but the swim generation is done, I think, through other circuits, and then it feeds back in a loop from behavior to reactivation of that circuit on the same side that the fish made the turn to. But in reality, it's of course a loop through the environment, because when the animal turns, that the body posture changes, it receives a lot of proprioceptive and mechanosensory feedback that gets fed back through mechanosensory sensory systems and so on. And so in reality, this loops through the environment back through sensory systems. And of course, it's also almost never the case that the environment of the fish is never featureless. So there'll be visual signals, vestibular signals, everything coming back in. So by simplifying the environment of the fish, we were able to kind of isolate this relatively small circuit motif, but in a real environment, it's going to be way more distributed. So it's really just an example of kind of focusing onto a small circuit, trying to understand it, but then knowing that later on we have to add back in all this other stuff, including eye movement. The Vatsala lab have found that eye movement also feeds into this circuit, and we know that visual signals come in, and we made some progress in understanding those as well. But yeah, there's always right, the tension between trying to understand a small circuit fairly precisely while also realizing that it's just part of one hugely interconnected network.
Thanks so much. Are there any questions? Okay, so we'll see you again tomorrow, Misha. Thanks for such a wonderful talk. Very nice.
Oh, it was a real pleasure. Thanks for the discussion.